Hi, my name is Sally. For as long as we can remember, A, T, G, and C make up our genetic code, and their combination stored all our information of life. In the recent 2 decades, scientists have tried to expand the existing codes and to find new alphabet for DNA. Along with the new letters, non-standard amino acids that have certain functions can now also be incorporated into proteins. Since they're at least the common ancestor of all life on Earth, from a tiny bacteria to a huge whale, genetic information has been stored in DNA with a 4-letter alphabet or the respective 4 different bases: adenine, thymine, cytosine, and guanine. A methylated thymine or uracil replaces thymine in RNA. Recently, 2 new groups, NAM and TPT3, that can substitute the bases have been found and synthesized, and their respective nucleotides can interact and bind with each other. Just like purines and pyrimidines are bound by hydrogen bonds, they are packed together with hydrophobic interactions. To fully expand the genetic language, we need much more than the new nucleotides and the new codons that they comprise. We also need non-standard amino acids to encode by the new codons. These non-canonical amino acids, or coincidentally NCAAs, are usually phenylalanine and tyrosine synthesized variants with specific purposes. In unique tRNA, that has the corresponding anticodon, which is able to recognize the new codon, along with the tRNA synthase that is able to charge the tRNA with the non-standard amino acid, are also required. There shouldn't be any crosstalk between the new tRNA-tRNA synthase combination with the existing ones. In March 2017, a group of scientists in San Diego reported their success on creating a semi-synthetic organism that is able to decode the new codon containing DNAM, DTPT3, or X and Y. They transformed E. coli with a plasmid containing genes for superfolded green fluorescent protein, or SFGFP, but its codon for the 151st residue is altered from TAG to AXC. The gene to encode the unique tRNA, tRNA-PYL, with the anticodon GYT is also included in the plasmid. The tRNA synthase for tRNA-PYL, PYLRS, is expressed in the separate plasmid. PYLRS can recognize and interact with PRK, which is a non-standard amino acid that resembles lysine, and together they can charge tRNA-PYL with PRK. The charged tRNA can then bind to the codon AXC using its anticodon, continuing the translation process. The semi-synthetic strain is named YZ3. As shown by the results, YZ3 displayed high GFP expression comparing with the strains with the natural codon TAG. And with the TAMRA dye, we can confirm that the non-standard amino acid PRK is incorporated into the protein. With the now expanded genetic language, we can have so much more control over any organisms. And for one, the semi-synthetic organism can help us produce novel therapeutic protein drugs with non-standard amino acids that has different functions. Since these organisms can't survive for long without the supply of artificial nucleotides, we don't need to worry much on contaminating the natural genetic pool. So in the near future, we can anticipate there are more and more of these new semi-synthetic lives that are able to access a broad range of form and functions. Thank you very much.